today's video, we will look at network motifs. I will introduce you to the concept of a motif in networks, which we have already seen a little while ago, but we will focus a little more on how do you identify motifs from a network, right, and that involves identifying a null model, randomization and so on. What are the different types of methods that are used to randomize a network, and what is the biological significance of these motifs. So, now let us look at motifs. So, what is a motif? We said it is some sort of a pattern, but we already defined it as an overrepresented subgraph, or rather a statistically significantly overrepresented subgraph in a network. So, not all subgraphs occur with the same frequency in any given network. You will find that some motifs are overrepresented, but overrepresented with respect to what? With respect to some null model, right. So, what becomes your null model? So, you can have different null models. You could think of a Barabasi-Albert null model, right, or you could think up of different null models. So, it depends upon what is the inference you want to draw. So, this is where I need you to recall your statistics, because this is a very statistical kind of argument, right. So, the common example I often give is when you are testing out a drug. Let us say you want to find out if paracetamol works against headache, right. How would you test if paracetamol works against headache? So, you first establish a group of patients, right. You probably want to actually establish a group that is very homogeneous, you know, it's got to have like 10 boys and 10 girls, maybe all between the age of 20 and 25 or whatever. It depends upon what is it that you want to test, but let us say you want to keep the things, the unknowns as narrow as possible, right. You do not want to have everybody in from the age of 10 to 60 or something like that. You want to keep a narrow control group and you want to have a narrow group which you are going to test the drug on. And how would you test it? What would you give the control group?
We will not have the drug but just the sugar pill.
Control.
Sugar pill.
Placebo.
So, what is a placebo?
Something that is not the drug.
That is not the drug, but does it have to have anything else?
It must look like a drug.
Maybe that is not important, but what is important is it should have everything in the tablet other than paracetamol. So, if you had some coloring agent, some other excipient that holds the tablet together, right, whatever else is there in the drug, maybe there is a color, right. So, even that color should be there, because then you cannot say that this color was not curing headache. See, finally paracetamol and the color are there in your drug, whereas paracetamol and color were not there in your control. You cannot still decouple the effect of paracetamol from the effect of the drug. Why I am giving you this example is, you can only answer the question based on the control that you set up. After this you find that 9 out of 10 people who were given paracetamol got cured of headache, and only 2 out of 10 people who had the placebo got cured of headache. You can now say with some confidence, you have to do a statistical test, that those who took paracetamol actually works against headache. So, in the same vein, here you have to set up the right control. What is it that you want to ask of this network? So, do you want to look at networks that have the same number of nodes? So, you could then say that in a network with thousands of nodes, I am very likely to find this triangle subgraph or this feed forward loop that I showed you yesterday. But is that what you want to do? Do you want to keep something else constant? You may want to keep the degree distribution constant, or you may want to keep the function constant if you are looking at a biological network, because then you can answer suppose you say you only keep the degree distribution constant and allowed for the function to change. It is not easy to measure the function of a network, but let us assume that you have a way to say what is the function of a network, whether it could be a metabolic network, then it is easy to say whether this network will grow on a particular nutrient source or not. So, then if you allow the network function to not change, you can now say that if you allow the network function to change, you can say that these motifs will be present in any functioning biological network, right. These motifs are necessary for function to happen in the first place. But if you establish the appropriate control, if you allow for function to not change and still your motif is enriched, then you can say that, oh, this motif seems to be very unique to my network, to the network that I am studying. It does not have to be a feature of all biological networks or all functional biological networks, but it seems to be a very special property of my network. This is usually something that you want to fish out of a data set or out of any given network. So, what is the statistical power that you have? So, it depends upon the null model you establish, the null, or the null hypothesis, alternate hypothesis, your good old statistical testing. You should try and brush up on your basic statistics. So, in this case what we want to study is how does my motif vary with respect to some null model. How do I now construct those null models? It depends upon the question. So, as always in systems biology, the modeling choice or the algorithm choice always depends. There is no one answer to what is it that you will use. It depends upon the question you want to answer. Do I want to identify what motifs are going to be present in this network, in biological networks or in my particular network? Depending upon the resolution of your question, the resolution of your model or your null model or your way to produce the null model is going to vary. So, let us start with a simple example. Let us say that I want to just keep the number of nodes and edges constant. What would you do? I would just generate an Erdos-Shreni model by saying n is this and p is e by n choose 2, and I would be able to create a graph with roughly the same number of nodes and edges. It will vary by a little, as we will see in the lab later today. It will vary by a little, but you will find that it is approximately represents the original network. But then what happens? You can only say that with respect to numbers now. For this size network, I expect so many triangles to be there, so many feed forward loops to be there. But then that is a very poor null model to compare. It is like comparing your paracetamol against maybe taking another bad a negative thing, right. But if you want to establish a more solid control, you would now say that okay, I want the degree distribution also to be constant. So, how would you rewire a network, or how would you create alternative networks? By the way, these null models are important in every scenario. If you want to define how good a community is, or in fact there is a definition of modularity that is based on these kinds of null models, right. So, modularity can actually be defined as the number of intra group edges minus rest of the edges, right, or the fraction of intra group edges minus the fraction of other edges. So, there are definitions of modularity that are based on this. So, let us say I want to keep degree distribution unaltered. How would I go about creating a null model? How do I create a new network? Or I do not want to create one network, I may want to generate a few thousand networks, right. This is very similar to the idea we saw yesterday, right. We said that this is going to be the distribution of some LWS, right, or rather LER, and my LWS comes here. So, this P will be much less than say 10 to the minus 5 or something like that. So, it is statistically significant, which means that I can say that LWS if I observe this. So, this actually we usually do not observe it for characteristic path length. So, characteristic path length is, whereas clustering coefficient, you will find that this actually happens. True. So, you have to but asking for degree distribution to be constant would not, you would not be going too specific. So, how would you create a new network that has the same degree distribution as the original network? You can actually resort to some sort of rewiring like your Watt-Strogatz. So, Watt-Strogatz was removing a particular node here and connecting it. So, in fact that is not the idea is rewiring, right. So, let us say this is your network, you have A, B and C, D, one way to rewire this would be, you now remove AB CD and make it AC BD, or you make it ADBC, right. This will obviously preserve the number of edges, or will it? Well, then the degree distribution. There is only one definition for degree distribution. So, it is n of k versus k. Will this graph change? Could be like this, it could be whichever way. See, what was, see, it is simple, right. So, what happens here after rewiring? It does not change, but is there a possible problem? I am always going to pick a pair and rewire them, and I can do multiple rewirings, that is not a problem. So, I will do enough rewiring so that I lose the memory of my original network. If I do one rewiring, I can't call it a random network, it is still a very close cousin of the original network we started off with. But if you do multiple rounds of rewiring, you can get a substantially different network. So, you could call it almost a random variant of the original network, and you repeat this process 1000 times, you will get a, you can compute a distribution of this sort. There is one small problem here. What if you already had an edge like this? You can't add a second edge between A and C, it does not work that way. So, this could be a problem. So, you may have to account for that, you may want to do a few more simulations, a few more iterations, wherein you ignore, if they already have an edge, do not do it, you try to rewire some other pair and so on. There is another way to do it.
So if I do that, then won't my degree distribution change?
So, A already, so here you have a problem. Here the thing is you can't add a second edge, here I can't rewire AC and BD now, right. So, I could, so let us say it is already connected like this, you have a problem. Now, I can't just say remove this and instead connect this. This means that I am going to add a second edge between C and D, which is not even legal, right. So, there is a problem there. So, you have to basically find another pair of ABC, another set of ABCD and repeat this exercise. But if in a sparse network, it is not going to be too much of a problem. One other way to go about is, think of these as threads and beads. Now, you just cut the edges here, leaving behind stubs, and now you tie any two threads together. This is what is the, what is the number of stubs tell you? Degree. So, you are not changing it. One problem, there is one problem here, can you think of it? Yeah, you do not want to be left up with left behind with this, right. Suppose you tied everything up, then you are left with having to tie a node to itself, right. But you can either permit that and have the appropriate null model, or basically reject it and try to come up with a different solution. So, all these are basically algorithms, right. So, while you have not done n number of rewirings, right, try one rewiring, if that rewiring is acceptable, change the graph to the newly obtained graph, repeat. This is the algorithm, and you can go about it either for this this kind of a rewiring or making these stubs and rewiring. Okay. So, last time we were looking at how we need to randomize a network, what are the methods that we would use to randomize a network, and what are the things you would keep constant. So, to identify motifs, one needs to identify all the subgraphs in the network, and you need to identify the statistically significantly overrepresented subgraphs. For this, you need multiple realizations of the original network, multiple random, what one calls bootstrapped variations of the original network. So, how would you randomize the network? You want to keep the number of nodes same, the number of edges same, as well as the degree distribution. For this, we discussed a couple of strategies to randomize the network, and following this procedure, the subgraphs that occur significantly more frequently in the real network compared to random will be designated as motifs. So what kind of motifs one might observe? So you might observe something that's known as a bifan, that's seen here, or a feed forward loop, a biparallel motif, or a feedback loop. Right? And you can have many more different kinds of motifs. One thing you can imagine is that the number of motifs exponentially increases with the size of the motif and of course the size of the network, the number of motifs that you can potentially find. Right? So Zhang and co-workers studied the yeast integrated network which contained several different interactions. So first was synthetic sick lethal interactions derived from a synthetic genetic array. So essentially this contains interactions between genes that when together deleted cause the organism to die, or at least cause it to fall sick, meaning grow slow or have some visible phenotypic damage. And then 40,000 odd protein sequence homology relationships, correlated mRNA expressions, protein interactions based on protein complexations, and a chip study to get some more regulatory interactions and so on. Finally giving you a huge network with about 6,000 nodes and 150,000 links. In this network, what were the motifs that could be identified? So this was one bunch of motifs. So if you can see, this is a feed forward loop, and what you find is they also have different edges here, right? They tag them as S, H, X, P, R and so on. So what you see here is the number of real networks were 4.7 into 10 to the 2, whereas the number of random was about 260, right? So 260 plus or minus 50. So it turns out that this is statistically significant, much higher than the random. So the first motif corresponds to the feed forward theme. There are other motifs corresponding to the co-pointing theme, regulonic complex, and so on and so forth. So all these you can see have different different significances, different patterns, and if you dig deeper, they also have different colors which correspond to different types of interactions. So there is a nice granular description of the motifs in this case. It's a very nice study that was published in the Journal of Biology in 2005.
Any functionality for the individual motif?
So all these motifs, I think they've gone back to show that they have some interesting functions and so on. So many of these motifs have, so if you see, all these are some ATP binding proteins and things like that. So they are able to capture the biology as well. These are modules.
Can I say the functionality of a motif, it governs the functionality of the network as number of motifs are large in a network?
Yes, it is one of the driving factors, right? The presence of a larger number of certain types of motifs may give you more robustness to the network and things like that. Certainly. Here you have more examples where they report the number of real motifs as well as the number found in random motifs, and there are the compensatory complexes, unclassified motifs, neighborhood clusters, so on and so forth. They basically identify different types of motifs in these networks. The one important thing to remember is that motifs are not independent, right? As you were just mentioning, motifs form clusters themselves, right? So they find that bifan motifs in E. coli transcription regulatory network form extended clusters. So many of these are bifans and they form extended clusters. To recap, a bifan looks like this. This is a bifan. And motif clusters seem to be common to all real networks. So in some sense we've understood very well how to identify motifs. So the first thing, it sounds simple, but when you get to practice you will see what are the challenges. So the first thing is you have a network, you identify all subgraphs in the network, and then you count them, and then you generate randomized realizations of the same network, count again, and then plot distributions and find out, or just do counts and averages and say, you can even do a parametric test if you want, right, to say that your number of motifs in the real network is statistically significant. But what is the challenge here?
computational.
Yeah. How do you enumerate all subgraphs in a network? It's going to be massive, millions, billions, I mean uncountable subgraphs, right? So then you have to resort to some interesting things like sampling subgraphs. So you try to sample parts of the network to find subgraphs and motifs, and you actually have intelligent methods that do random sampling based on subgraph concentration. And one also is very interested in studying how motifs evolve and are there conserved motifs across species, across kingdoms of life, across organisms, across different types of networks. You could have a co-expression network versus a metabolic network versus a gene regulatory network, and so on. So Uri alone has this very classic textbook on systems biology which covers a lot of information on motifs and networks. So I hope you got a good glimpse of what motifs are, some concepts related to randomization, statistical hypothesis testing in terms of identifying motifs in a network, as well as how these motifs are embedded in biological networks. I really invite you to go and read a little more about motifs in the book by Uri alone I just showed. And in the next video, we will move towards a lab session wherein I will introduce you to cytoscape, talk to you about some concepts of the underlying the string database, as well as how do you load and visualize networks and use this very interesting cytoscape tool called Network Analyzer.